I came in, I'm wearing the same thing. I'm doing a little short sort of live lecture capture for the last part of our lecture on action potential. So I know some of you had to leave right when the video was playing, the slide right before this, and we were kind of right up at the end. Ooh, I'm not gonna play it again. But that is at the end of the last little segment of video, and so I'm doing this one just to finish this last part of the material of action potential. So in conducting an action potential, we went over all sort of the ion channels, the ions, and the flow of depolarization, repolarization, hyperpolarization. This goes over exactly how that's happening along the axon. So showing that there are sort of discrete action potentials, if you will, moving along in sequence sort of along the axon. Okay, so in an unmyelinated axon, so it doesn't have a lot of that myelin sheath, we're just looking right at the axon. Sodium rushes in, spreads to the next area, opens sodium channels, voltage-gated sodium channels because of the positive charge. Sodium comes in. Right behind that, potassium flows out. So now this triggers yet more voltage-gated sodium channels to open in the next adjacent patch of membrane. Sodium rushes in, and so on, and moves down. And then potassium goes out right behind it. So in this way, action potentials sort of overlapping are zipping down the axon of that neuron. Okay, so showing and sort of going step by step, that's how it flows along. And move forward. Okay, now, if that were the case and all the neurons and axons are unmyelinated, so they're just axon, there would be a lot of conduction loss. So there would be charges that are leaking out, it would take, there's resistance inside of the cells, there would be diffusion of charge and resistance of charge and sort of ions leaking out. They wouldn't go in a nice fast trajectory down. So the way that neurons have compensated for this loss of quick charge flow is by to wrap their axons, of course, in support cells. And in the case of the peripheral nervous system, a lot of neurons are wrapped in Schwann cells where they find the neuron axons and they wrap themselves in plasma membrane loaded with layers of myelin, which is a lipid, inside of the cell, and it wraps almost like a flat pancake, like all the way around these given segments. So all the way down the axon, for a myelinated neuron, they're covered in myelin sheath, which is these layers of plasma membrane and myelin that are packed inside of these support cells. So this is one, this is one, this is one, all the way along. There are gaps or nodes between those Schwann cells, and those are known as the nodes of Ranvier. So the important part about all of this creates almost like an insulation or a cable casing that goes around the neuron axon, preventing leakage of ions out of that axon, and actually propagates action potentials more quickly from the top to the bottom of the axon. So what this creates is a setup. You only have ion channels exposed at the nodes of Ranvier. So this is where ion channels, and we talked about this, ion channels are clustered at those nodes of Ranvier, which means current rushes in, current rushes out. And that current has nowhere to go. So the sodium comes in, it doesn't go back out, it actually moves down to the next node, opens those voltage-gated sodium channels, moves down to the next node. So you can see that this propagation of action potentials, if the neuron is myelinated, greatly speeds up the AP conduction. So it actually jumps from one node to the next. It's called saltatory conduction, as it's listed right at the top right there. That means to jump. Kangaroos, they have saltatory mobility, right? They jump around. So this causes charge and action potential to jump from one node to the next. Come in, spread to the next one, come in, spread to the next one. So if an axon is very large, like the diameter of the axon itself is very large, and it has this whole casing of myelin sheath around it, it's going to greatly speed up action potential propagation all the way down the neuron. And some of the neurons in the body, actually, the ones that activate muscles, for example, like skeletal muscle, are really, really quick. You can react, you can flinch, you can have reflexes, you can jump, dive, dance, do all those things very, very fast. So these action potentials just zip like lightning right down those neurons. All right, application question, something to think about. And I'll just put this on here and I'll let you write it down individually. and try to draw it and write it just like we do the other application questions. And then what I'll do, I think, in all of these lecture captures, I'll go over the previous application questions so I can kind of go through what I was looking for and kind of the best way to answer these. So you can do this on your own, of course, or you can work with your roommate or a study mate or whoever, probably by yourself. Many of you are going to be doing some of these by yourself. So there's a drug and an axon as an irreversible inhibitor of voltage-gated sodium channels. Now you know what voltage-gated sodium channels are and how they work, where they're important. What would happen if you applied that drug, that's an inhibitor of voltage-gated NA channels, to your neurons in culture in the dish, so you're studying them in the lab, and then you stimulated them to fire an action potential. So you treat them with the drug, you bathe them in the drug, then you try to stimulate them with an electrode to fire an action potential. Draw the oscilloscope membrane potential trace that would result from this experiment or this scenario, okay? So I would set it up. I won't do it, I'll just show you how to set up time, membrane potential in millivolts. And I'm going to start at my resting membrane potential, right? My minus 70, somewhere around there. I'm going to go here, and then I'm going to say, okay, so this is treated with the drug, so I'm putting the drug on, bathing the neurons with the drug, and then what is going to happen when I stimulate those cells to fire an action potential? What exactly is that going to do to the typical neuron trace of when we have resting membrane potential, then an action potential, and then recovery, right? So I want you to draw that. Same thing, but a different scenario. Another drug is a competitive inhibitor of the sodium-potassium ATPase. So this one blocks the NAK-ATPase. What would this do to your neurons if you applied the drug and then tried to stimulate them to fire an action potential? Same scenario. What I want you to do is draw that out and give a rationale for why. Why that blocking of that specific channel or pump is going to create the results that you draw and what you expect to see, okay? So what would this do to your neurons if you applied the drug and then stimulated them to fire an action potential? Draw the oscilloscope membrane potential neuron trace. Same thing, draw. Okay, so two of those. I want you to kind of work through those on your own. You may not get them exactly right, but as long as you kind of do it and you have a reason for why, when you knock out that and when you knock out that, that is good if you can think along those lines. Okay, going on to this last part. Oh, I don't know, let's see. Oh, it works. Okay, so we're going to step through each little part of this. This is showing action potential in a big cartoony fat neuron. If I step through, step by step, you can play this in your own computers with the PowerPoint mode presentation on, and you can do all this. It blows it up. We've got electrodes. Okay, so we're monitoring charge. And if there's a positive charge that comes into the cell, or we stimulate that neuron to fire, something that's called an excitatory postsynaptic potential, or an EPSP. That means positive charge is coming into the cell, coming in, and if enough comes in, you might be reaching that threshold. Remember, we referred to threshold at about minus 55, right? This is the axon hillock, end of the cell body, beginning of the axon. This is a myelin sheath, okay? And this is exposed nodes of Ranvier. Now, if we keep it going, step it forward, sodium will rush in. If it reaches that threshold, and it will fire an action potential. Now notice, enough positive current came in here, positive ions, current, same thing, right? Gets to that membrane potential of minus 55, opens voltage-gated sodium channels, sodium rushes in, and look, myelin sheath, Schwann cell or oligodendrocyte myelin sheath, prevents that leakage or loss of charge, makes that sodium rush down to the next node. Coming in, rushing. So just by doing that, there's 100 or 1,000-fold increase in how fast that charge is actually going. So here's the next node of Ranvier, and you can step through there, and boom, it opens those sodium channels, sodium rushed in there, and then boom, propagates past the next set of axon membrane. It goes all the way to the next node, which would be like over here. And notice behind this, voltage-gated potassium channels, when the voltage goes up, then they open and potassium leaves. So each one of these is almost like an independent action potential happening at each node, and it's jumping from node to node. So the vink, next one, the vink, and it would continue doing that. If I step through further, boom, it's going to finish. Potassium's going to go out, sodium comes in, jumps down, potassium clears. So you're going to get a bunch of these, boom, 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 all the way down. And that's how action potentials propagate so quickly down myelinated axons. All right, I think that's the end of that one. There might be one more step. Oh, one more just going along. Okay, wow, I messed that up with chicken scratch. Sorry. Okay. I will erase this. Get us back to regular screen. Okay. And this brings me to all or none law. Okay, so we've talked about this and I've said things are graded. If it's graded, it means a certain amount has to build up to reach that threshold. If it doesn't, and it doesn't, it's not going to fire. Action potentials are all or none. So if they reach threshold and they open those voltage-gated sodium channels, that neuron is going to fire an action potential. Once thresholds reach the AP is going to happen. We just refer to that in sort of cell biology as the all or none law, or in physiology as the all or none law. So the size of the stimulus is not going to affect the size of the action potential. Action potential is always going to reach about 30 millivolts, depending on what type of cell it is or what type of neuron. Always going to reach about there. So in other words, if it gets a really strong stimulus, a lot of neurotransmitter dumped on it, or I'm injecting a whole bunch of charge, positive charge, and really zapping it, doesn't matter how big that stimulus is, it's always going to look the same. It's always going to go up and then come down. It's always going to have the same magnitude. The size of the stimulus also is not going to affect the action potential duration. That means the length of time that AP takes to fire is always going to be the same. Different neurons, based on their myelination, can fire at different speeds. But in that individual cell, a given action potential is going to be the same duration and same magnitude, no matter how strong of a stimulus or how much neurotransmitter that cell is exposed to. But how it's affected is a really strong stimulus or a lot of stimuli at the same time, so a lot of neurotransmitters or synapses affecting that one cell at the same time will actually make action potentials fire more quickly or more frequently. So stimulus intensity converts into action potential frequency, okay? How strong the stimulus is or how much of it is provided to that cell is going to determine how fast that cell fires, how fast it sends off these action potentials. All right, so if we look at this, and some of this is a little kind of review. We've talked about stuff like this a little bit more in the past. So all or none law, action potentials. You give a weak stimulus, it's not enough, it doesn't reach threshold, it's not going to fire. Stimuli, we can give single quick shocks, electrical current, and basically we can get action potential, we can give an even stronger shock, action potential. So based on these single quick shocks, whether they're weak or really strong, the magnitude is always going to be the same in an action potential. It's going to get up to a certain millivolts and it's going to go back down. That does not change. What changes is if we give longer stimuli, like we turn them on and off, dump on a bunch of neurotransmitter and then remove it, or put in electrical current and then stop it. If we do a weak stimulus, we don't get action potentials because threshold is not reached. We do an intermediate or a big enough stimulus, that threshold is actually reached, and we'll get a frequency of firing that's like boom, 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 boom. So that neuron is kind of firing at a slow, steady rate. However, if the stimulus is ramped up, AKA there's lots of neurons releasing neurotransmitters on that one neuron, enough to where threshold is reached and it's a lot of stimuli, that will cause enough positive charge to rush in there, so much that the action potentials will fire and then reset and fire, and they'll do it really, really quickly. So the intensity of the stimulus, or how much neurotransmitter that cell is exposed to, will determine not the magnitude, not the duration of how long it takes for an AP to fire, but the frequency of APs going off. So in this case, with a great stimulus applied, it ends up going brrr, super fast. And there's neurons in your brain and body that actually do exactly this. So stimulus intensity is coded by action potential frequency, okay? They change their speed of firing. That is all, that's it, that's the last section of this. So the next lecture capture I'm gonna do in like a complete chunk. It's not gonna be broken up because we've had the little technological phone stuff today. Next lecture capture is gonna be kind of all in one piece. And I'm gonna, of course, provide you the links. So you guys just have to open the links and watch it and take notes and then jot down any questions that you have. Okay, see you all later. Stay well, stay happy, stay balanced, stay safe, and I will see you later. Okay, bye.